All right. Hi, everyone. Excited to be here. My name is Ian. I'm going to be presenting on IsoCLR, Contrastive Learning for RNA Foundation Models. This work was co-authored by Philip Fradkin, who is in figure one over there, along with our collaborators, Karen, and our supervisors, Quaid, Bo, Brendan, and Leo. All right, so IsoCLR is an RNA foundation model, but let's talk about what foundation models are. So recently, inspired by successes in natural language processing, folks have seen there's a lot of unlabeled data sets of genomic sequences. Can we just throw a deep learning model onto it and see what comes out? Basically, so these foundation models are large models that are trained on huge unlabeled data sets of genomic sequences. And the main purpose is to learn a representation of these sequences that are useful for a variety of downstream biological tasks. So these foundation models undergo first a pre-training phase. So in this phase, we take our large data set of sequences and we apply random perturbations onto the sequence. And the only objective here for the foundation model is to reconstruct those original sequences without the perturbations. So it's quite a simple objective. This is known as self-supervised learning, but as we've seen in other domains, this actually allows the model to learn the underlying structure and semantics of the domain, usually. And from that, we get a useful latent representation for our genomic sequences. Using that useful representation, we can do transfer learning. So here we have a fine-tuning data set. This is a usually smaller data set where we actually have labels for our sequences, and then we're able to fine-tune our foundation model using those pre-trained weights for the prediction task. And the idea here is that those weights and the associated latent representation allows for better convergence and better generalization. So just for some concrete examples, if we train, for example, on unlabeled RNA sequences, we might take some mRNA sequences and we want to predict, for example, mean ribosome load, or the secondary structure for non-coding RNA sequences, or in the DNA world, we might want to, for example, classify these sequences. Okay, so we use a self-supervised learning objective. There are two main ones that people use these days. The first of which is mass language modeling. Here, we just take a sequence, randomly mask out some tokens or nucleotides, and we ask the model to fill in the blanks from the surrounding nucleotide context. We also have next token prediction, where we just take a sequence, ask the model to predict the next token. So relatively simple. And because of that, there's actually been a lot of models out there that have been trained using these self-supervised methods that are genomic foundation models. So this is a non-exhaustive list. It seems like there's a new one coming out every week. But these include DNA BERT, nucleotide transformer, RNA FM, and hyena DNA. And I'm not including things like actual fully supervised genomic foundation models like Informer here. But basically, these models are trained on typically actually DNA, but there's a diversity of data sets. One commonality is that they use the transformer architecture for their sequence modeling. As we all know, transformers are very powerful, but they do suffer from a limitation that they're quite memory intensive, meaning that we can only input about 1,000 nucleotides of context for a transcript, which is much shorter than the average RNA that we might want to model. So we have to resort to things like chunking it up. It's not ideal. Hyena DNA uses something called the hyena hierarchy, which is closely related to what IsoCLR, our model, will be using. And because of some methodological advancements, we're able to model a much longer context window. We'll get back to that later. Okay, so let's take a step back. Seems like there's a lot of models actually already. Why do we need RNA foundation models? And why do we even need IsoCLR to begin with? So this is kind of preaching to the choir, but first of all, I think we all agree that RNA is very important. So if we are able to model RNA properties, we could use this for many biomedical applications. For example, if we are able to predict RNA half-life or stability from sequence, we could use this for like mRNA vaccine design. But if we looked at those two self-supervised objectives from before, there was nothing actually specific to RNA about it, right? So they're typically trained on DNA, and RNA transcripts are a subset of the genomic DNA. So you could just say, could we just use a DNA foundation model for RNA tasks? And spoiler alert, the answer is no. Hopefully, we'll show in the results later. And here's why we think why. These DNA foundation models tend to randomly mask out tokens from the entire genomic DNA, right? And we also know that information is not uniformly distributed within the genome. That means that if I was to just pick a random position, it's probably not in an exon. And if it's not in an exon, then it's probably just random noise. So these models, we're arguing, basically just predict random noise. They're not very useful for genomic tasks other than a small subset. And yeah, they're predicting noise. So this is why we introduce IsoCLR. IsoCLR is a RNA foundation model that is trained on mature RNA transcripts to focus on high signal regions. And we adopt a novel contrastive learning objective based on biological annotations to more incorporate biological information into our foundation model. We also use the Mamba architecture for its sequence modeling. So Mamba is this fancy new sequence modeling thing that's related to the hyena thing I was talking about. Basically, it's a deep state space model that is represented by those equations, but there's a lot of magic that happens under the hood that I won't go into today. The overall takeaway is that basically it's really efficient at modeling long sequences. So it scales with an L log L complexity, where L is the sequence length compared to the transformer's L squared complexity. Basically, this means on a consumer GPU, you're able to fit entire RNA transcripts into a single inference pass. Okay, so we just said that we introduced new contrastive learning loss. What is it? So we'll go into it here. These self-supervised learning objectives aim to learn a good latent representation. So we're just going to say, let's maximize the latent similarity between splice isoforms and orthologs with any reference transcript. So we'll denote each transcript's latent representation in this big model as a dot here. And before the model training, our latent space will look something like this, right? Our transcripts are just uniformly randomly distributed in the latent space. And our objective during training will be to pull these orthologs and splice isoforms of a reference transcript closer together and then push unrelated transcripts further apart. And by doing this, we'll generate a structured latent space that is useful for downstream tasks. We will accomplish this using something called the decoupled contrastive learning loss. And just to quickly break this down, for computational efficiency reasons, we'll only look at 2 pairs of transcripts per single iteration. So we've labeled the positive pair transcripts. So these are the transcripts that are related, like splice isoforms or orthologs, in blue here. And we're going to maximize their similarity. And meanwhile, the unrelated transcripts are shown in red in this loss function. So we'll consider similarity using cosine similarity. That's shown in these angle brackets in these equations. And basically, the top line is saying, yeah, let's maximize the similarity between the positive pairs and minimize the similarity between positive pairs and unrelated pairs. Okay, so that was our loss function. How are we actually going to pre-train our IsoCLR model? So we'll take our pre-RNAs, right? And then these will be spliced down to splice isoforms based on some, and we'll get this information from some sort of annotation. Either way, we'll get our splice isoforms, and we'll call this one mature transcript set. We'll also consider orthologous genes another possible source of transcript sets. So we'll collect a giant data set of all these transcripts and their associated transcript sets. We'll just randomly sample them during training to pull out some transcript sets from the data set. And then from each transcript set, we will sample each of our positive pairs, since we only want to look at 2 at a time. Animations are super in these days. Okay, and then these sequences will be embedded using our Mamba model. And then using the average of these sequence embeddings, we will compute our contrastive loss, and the whole model will be trained like this. The actual sequence annotations we've obtained from GenCode and RefSeq for human, mice, and 8 other species. We've also done this hacky thing where we say that transcripts that share gene names are orthologs. Of course, they're not actually orthologs most of the cases, but if we don't do this, it doesn't actually work for some reason. And then finally, we'll look at real orthologs from Project Sinumia. So Project Sinumia is really cool. They've done alignments from all eutherian mammals to human and mice. And we'll consider this as another data source. Okay, in all, we wound up with 49 million transcripts and 850 million positive pair interactions between these transcripts to use to train our model. Just some final notes on our data processing. We treat the RNA sequence as a one-hot encoding, and we also include splice site and reading frame information. And we also do, also randomly mask out the sequence for further augmentation regularization. Okay, at this point, let's assume our model has been trained. We've wasted tons of GPU hours. Let's convince you guys that this actually works. So we'll look at a couple of RNA property prediction tasks here. The first of which is RNA half-life. This is just the decay rate of mRNA. We'll also look at mean ribosome load. So this is the number of the ribosomes that are bound to an RNA during translation. We'll try to classify protein subcellular localization of the actual final protein product, as well as this gene ontology term. Okay, so those were the property tasks we're going to consider. We'll also consider them in 2 contexts, the first of which is linear probing. Here, we just take the model immediately after pre-training. We'll freeze the weights, and we'll just train a linear classifier on top of it. This basically just evaluates the quality of the latent space in terms of linear separability. We'll also perform full-on fine-tuning to basically see how useful this model is in the transfer learning context, to see how well we can do. Okay, and just one final note, we've kept homologous transcripts within the same data split for all of our tasks to make sure that there's no leakage in that form. Okay, so here we're showing the results of our model on the linear probing context. Each of these plots represents one of the property prediction tasks we're modeling. For the regression task, we've shown the Pearson correlation, and for the classification task, we've shown the area under the receiver operator curve. So we've classified each of these performance results based on models. So IsoCLR is shown in blue. The reason why there are multiple models per sort of group is because the authors often publish multiple model sizes based on parameter count. But basically, we clearly see IsoCLR outperforming all the other existing self-supervised genomic foundation models, which is really exciting for us. And we see that these results also extend in the fine-tuning context. So here, we have IsoCLR again in blue. Supervised Mamba is our same architecture, but without the pre-training step. So instead of using the weights that have been learned from this, minimizing self-supervised, maximizing splice isoform similarity, we just use random weights. We see that doesn't perform as well. We also compare against Saluki, which is a dilated convolutional neural network, I think, trained on RNA half-life, as well as hyena DNA, which we talked about previously. Okay, but what we're really excited about actually is the application of IsoCLR in small data context. So what this plot is showing is the performance of the model on RNA half-life for fine-tuning. On the left, we use the entire data set, but as we move to the right, we use a smaller and smaller fraction of the data set to train our prediction task. And so what we're showing here, what we're seeing here is that IsoCLR is actually able to retain 73% of the original performance using only 45 data points. So we think that our model could be really useful in these contexts where we have small-scale biological experiments and you're able to sort of extrapolate those results very efficiently. Okay, just some final slides on the model latent space visualization. Here, each transcript is a dot. We've done a t-SNE plot, and we've colored it based on transcript type. And we see, I think, nice separation there. I think one of the things that people always say to us when we present this model is like, well, the whole point of splice isoforms is that they are a little bit different, right? It doesn't make sense to bring them closer together in space. So I think one thing to realize is that even though the splice isoforms are different, they're still more similar to each other than completely different transcripts. And we also do see that the model learns the difference between different splice isoforms. So that's what this plot on the lower right is showing. This is a splice isoforms for one pre-RNA. And then we're basically showing the color is the distance in the latent space between the different splice isoforms, where the brighter color means it's closer. We see basically that every one of these splice isoforms is closely related to the protein coding isoform, but not to each other. So that's promising. Okay, and we've also made a lot of decisions in our model. So this is just showing some of the results of our data set construction. For example, on the right, how important was including the splice isoforms in our pre-training objective, or how important were the orthologs? And basically, we're showing that each of our decisions here do have a positive impact. And again, another heat map showing the combinations of the different augmentations that we've chosen. This is basically the key takeaway is that the actual masking step was quite important to preserving performance. But this is, I think, a cool area of exploration here because within the orthology we have like sort of a tree of life, and it could be interesting to see what the different contributions of using different clades are here too. Okay, so yeah, basically what we just talked about is IsoCLR, which is a RNA foundation model trained using contrastive learning. It performs well. Its latent space is well structured for RNA property prediction tasks, and we show that fine-tuning outperforms supervised performance on downstream data sets, especially when we have a small fine-tuning data set. And we think that this contrastive learning objective is also widely applicable to existing genomic foundation models. And it's quite cool because, as we saw with the previous self-supervised learning methods, it was quite biology agnostic, and here we're integrating biological annotations, and we think that's pretty cool. And hopefully we'll get the code and model weights out soon so everyone can try it. Yeah, with that, I'd like to give thanks to all my lovely lab members as well as Phil's lab members. And yeah, I'm excited to take any questions. Thank you.
Hi, so very impressive, and I'm excited and looking forward to using the weights and the model. I was wondering if you showed us 5 scenarios where the model did very well in improving the prediction performance in the task. I wonder if there were any tasks where the model didn't improve prediction performance or even deteriorated performance, because from my experience with DNA BERT and other competitors, they don't always do that magic. It could be that the signal is very local. It could be that you have many data points labeled that you don't need it, or it's a synthetic environment that they don't learn something from the genome. So can you comment on that?
Yeah, thank you for that question. So yeah, of course, this is not magic. I usually like putting a limitation slide up here so I can talk about that. I think one thing is that we might have been overzealous in removing these mass language modeling objectives, because that's really good at local tasks, and whereas this is sort of more good at global tasks, like what is the property of the entire transcript. But if you were to ask this to do like sort of let me predict whether an RNA binding protein actually binds here, we're not quite sure it would do as well on that. Basically, it just doesn't have good information on locality, but we are working on follow-ups to sort of address that scenario. I think that would be the main limitation, just the fact that it does better on global tasks.
Hey there, thank you for your presentation, really nice. Actually, I'm working in something similar here, like RNA foundation model, slightly different. One question here, you showed something about the embeddings of isoforms of genes. Did you test your model? It's not working. 1, 2, 3, yes, that's it. So did you test your model on for example, the half-life that you showed? If there is an example that isoforms of a gene have different labels, have you checked them? Because your model will give you similar embeddings for the isoform of a gene, right? And if the task that you're trying to predict is actually, if you want to gather or talk about the differences between isoforms of a gene, does your model able to do that?
So I think this plot here is showing that the model does kind of learn some differences between isoforms in the gene just without any instruction, but obviously our objective is trying to maximize the similarity. So I guess it might not necessarily learn the differences between isoforms. We haven't really evaluated it, basically, I guess is my answer to that.
I see. So then what's the point of having here, when we increase the resolution from gene to isoform, we're interested in seeing the differences between isoforms. Then what's the point here that you put all of these transcripts together? You know what I mean? If it's not.
Yeah, it's just like a source of augmentation, basically, is how I would describe it. So I think, well, you when you're constructing a lane space, I guess you want to understand what the points are in that lane space so you can perform good interpolation between the points, right? So if you say that one transcript is similar to another's transcript properties, including these isoforms basically allows you to construct a better lane space so that this interpolation can be done efficiently. That's what we're most focused on right now. But I would say I'm not saying that it wouldn't be able to understand the difference between isoforms. I think we just haven't necessarily evaluated it yet, so I can't say anything to that point.
I see. Thank you.